Welcome to the session. In this session, we are going to study about the mammalian cell and its organelles. So, cell is the smallest structural and functional unit of the body, which is the basic unit or building blocks of the body. There are 100 trillions of cells in our body. Each cell has distinguished features, whereas there are some common characteristic features in the cell which we are going to study in this session. Antony von Leeuwenhoek was the one who discovered microscope. After his discovery, Robert Hooke focused some tissue under microscope and he saw some room-like structures. He named it as cells. So Robert Hooke was the person who discovered cell. Antony von Leeuwenhoek discovered microscope. So these two scientists, Schleiden and Swan, they proposed a theory which is called cell theory, which states that all living organisms are composed of cell or cells, and all the cells arises from the pre-existing cells. So this is cell theory, which was proposed by Schleiden and Swan. So how the cell is organized in our body? So there are 100 trillions of cells. So how they are arranged? So there are cells, and group of cells forms a tissue. Here we have an example of smooth muscle cell, and group of smooth muscle cells together forms smooth muscle bundle, smooth muscle tissue, and group of muscles together forms group of muscle tissue, which means here we have an example of urinary bladder, which is made up of smooth muscle as well as skeletal muscle, and group of tissues together called organ. And here we have example for urinary bladder, and group of organs together form system. Here we have an example which is showing the urinary system or excretory system. So to complete this, how cell is organized? There are cells, group of cells together forms tissue, group of tissues together forms organ, group of organs together forms system, which is performing one function. Here for this example, urinary system performs, what is the function of urinary system? Yes, it is the formation of urine and removal of waste products from the body. So that is how the cell system is organized in our body. What is the basic structure of cell? So in our body, the cell has three important components. So we can see there is a cell membrane, and we have cytoplasm, which is having cell organelles, cytoskeleton, and even some cells have inclusion bodies. In the center of the cell, we have nucleus, which is covered by nuclear membrane. Inside the nuclear membrane, we have nucleoplasm, which is the cytoplasm of nucleus, and we have nucleoli. So these are the basic structures which we are going to study detailing. So first we will start with the cell membrane. Cell membrane, which is also called as plasma membrane, it is 7 to 10 nanometer in thickness. We can also say 70 to 100 Armstrongs in thickness. It's a bilipid layer, and it is a protective sheath which is enveloping the cell body. What is the function of plasma membrane? The first function is, we can say it is a protective sheath which is protecting the contents of the cell. And the second function is it separates the contents of the cell with the external environment, and it also controls the exchange of materials between the intracellular and extracellular fluid. One more function is, it is the one which provides the selective permeability, which means it allows some substances into the cell, and for some substances it acts as a barrier. So that is why it is also called as semi-permeable layer. Cell membrane is a semi-permeable layer. It is mainly due to the presence of lipids. So if we see the composition of cell membrane, it is mainly composed of proteins. So the major components of cell membrane is proteins, which is 55%, and then lipids, which is 40%, and carbohydrates, which is 5%. So the structure of cell membrane is explained by a hypothesis which is called as fluid mosaic model, which is the most widely accepted hypothesis, which was proposed by Singer and Nicholson in the year 1972. According to this hypothesis, so here we have a lipid molecules which are arranged as a continuous structures, and this forms the fluid component. And as a discontinuous structures, we have protein molecules which are present in the cell membrane, which is floating in the lipid layer of the cell membrane. So this forms the mosaic component of the fluid mosaic model. So this was the hypothesis which is widely accepted, and this is called fluid mosaic model hypothesis. And so we said the composition of cell membrane is lipids, which are the major components, and then proteins, which is 40%, then carbohydrates, which is 5%. So we'll study one by one detailing. So the lipids, there are three different types of lipids which are mainly present in the cell membrane: phospholipids, glycolipids, and then cholesterol. So these are the three important types of lipids which are seen in the cell membrane. Phospholipids are the most abundant lipids in the cell membrane. So which is the major component of cell membrane? That is proteins. But among lipids, we can say which is the major component of lipid which is seen in the cell membrane? That is phospholipids. So the phospholipids are arranged like so here we can see a structure which is showing the head end and the tail end. So this is the basic structure of the lipid molecule where we have head end and tail end. The head end is composed of phosphate moiety of phospholipid or hydroxyl radical of cholesterol. And this head end is the positive end and it is soluble in water. So that is why it is called as polar end or hydrophilic end. And the tail end consists of two chains of fatty acids or steroid radical of cholesterol. It is quite soluble in water, and it is negative charge. So that is why it is also called as non-polar end and hydrophobic end. So non-polar hydrophobic tail ends are directed towards the center of the membrane, whereas the polar hydrophilic end are directed towards the either side of the cell membrane. So what is the functional significance of this lipid bilayer? Lipid bilayer of the cell membrane makes it as a semi-permeable membrane. So here we can see the structure. The lipid molecules are arranged as a bilipid layer. So cell membrane is a bilipid layer. So here we have lipid molecules which are having head end and tail end. The head end, as I told you, it is a polar end. It is facing the outer side of the membrane, whereas the tail ends are facing towards the center of the membrane. And this, because of the presence of lipids in the cell membrane, this membrane is a semi-permeable membrane which allows only lipid molecules to pass through easily, whereas water-soluble molecule and lipid insoluble substances cannot pass easily through the cell membrane. Even they can pass through the cell membrane. We are going to study separately how the different kind of molecule transports across the cell membrane as a separate topic. So just remember, cell membrane is a bilipid layer which is semi-permeable layer. So proteins in the cell membrane. There are three different types of proteins in the cell membrane. Proteins which are combined along with lipids or carbohydrates, which are lipoproteins and glycoproteins. And there are two more different types of proteins which are peripheral proteins and integral proteins. So lipoproteins, which means proteins which are attached to the lipids, and glycoproteins are proteins which are attached with the carbohydrates. The lipoproteins can act as enzymes and ion channels. Glycoproteins act as receptor for hormones and neurotransmitters. And peripheral proteins are again two types: intrinsic proteins and extrinsic proteins based on their presence. So intrinsic proteins are present on the inner surface of the membrane, whereas extrinsic proteins are present on the outer surface of the membrane. These intrinsic proteins, they act as enzymes or anchoring proteins, whereas extrinsic proteins act as cell adhesion molecules. Transmembrane proteins, how they are present? Transmembrane proteins are present across the cell membrane, and they even act as channel proteins, carrier proteins, and receptors, and they also act as enzymes. So how the carbohydrates are present in the cell membrane? Carbohydrates are present by attaching with the proteins, they are called as glycoproteins, or they are attached with the lipids, which are called as glycolipids. The main function of carbohydrates is they form a thin membrane just below the cell membrane, which is called as glycocalyx, which helps to fix the cells to one another. So they also provide the negativity to the cell membrane, which prevents the negatively charged molecules to move out of the cell, and they also act as receptors. So this is about the cell membrane. So next we will see the cytoplasm and the cell organelles. So what is cytoplasm? It is a jelly-like fluid portion which is also called as cytosol. And the cytoplasmic structures are divided into cell organelles, cytoplasmic organelles, inclusion bodies and cytoskeleton. So here we have some of the cell organelles. So these cell organelles are permanent components which will have limiting membrane, and they mainly participate in cellular metabolic activity. So you have seen these cell organelles: mitochondria, endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, peroxisomes, centrosomes and centriole. All these are cell organelles. So we can name the cell organelles: mitochondria, endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, peroxisomes, centrosomes and centriole. So mitochondria is the powerhouse of the cell because it is the one which produces ATP and stores ATP. So mitochondria is self-replicative, where it has its own DNA. Mitochondria has its own DNA, similar like in the nucleus. It has some differences. The nuclear DNA and mitochondrial DNA has some differences, whereas mitochondria has its own DNA because it is self-replicative. Mitochondria is mainly, the genes of mitochondria, which is mainly derived from ovum, so it is maternal inheritance. Mitochondria is also plays very important role in the apoptosis. What is apoptosis? Which is a programmed cell death. So here we can see the structure of the mitochondria. Mitochondria is usually oval shaped or sausage shaped structure. It has two membranes, the outer membrane which is smooth, and we have inner membrane which is thrown into folds. The inner membrane is called cristae, and the matrix of this cristae has various enzymes which are participating in the various metabolic reactions like TCA cycle. So we can say one example, that is TCA cycle. The enzymes which are involved in TCA cycle is present in the matrix of the mitochondria. So here we can see the lollipop like globular heads which stores ATP. So that is why it is also called as powerhouse of the cell. So the next cell organelle is endoplasmic reticulum. So what is endoplasmic reticulum? It is a network of tubular and flat vesicular structures. We can see there are flat vesicular structures and tubular like structures. So these collection of tubular and flat vesicular structures are called as endoplasmic reticulum. It is called as synthetic machinery of cell, or it is the quality control of cell. Why it is called as synthetic machinery of cell? The main function of endoplasmic reticulum is it is the one which synthesizes proteins, lipids and steroids. So here you can see there are two different types of endoplasmic reticulum. So granular endoplasmic reticulum and agranular endoplasmic reticulum. So there is other names. Granular endoplasmic reticulum is also called as rough endoplasmic reticulum, whereas agranular endoplasmic reticulum is also called as smooth endoplasmic reticulum. This rough endoplasmic reticulum, on its surface it has ribosomes, whereas smooth endoplasmic reticulum, it does not have ribosomes on its surface. That is why it is smooth. So we will study what are the major differences between this rough endoplasmic reticulum and smooth endoplasmic reticulum. So as we see in the earlier picture, rough endoplasmic reticulum has ribosomes on its surface, whereas in smooth endoplasmic reticulum there is devoid of ribosomes on its surface. So what is the function of rough endoplasmic reticulum? Since it has ribosomes on its surface, it mainly involved in the synthesis of proteins, whereas smooth endoplasmic reticulum synthesizes lipids, steroids, and it is also involved in detoxification of various drugs or even toxins. So in which cells rough endoplasmic reticulum will be more numerous? It is obvious. Any cell which is involved in protein synthesis will have more number of rough endoplasmic reticulum. So the same thing goes here. The cells which will involved in the lipid and steroid synthesis will have more numerous smooth endoplasmic reticulum. So here we have an examples like Nissl granules of nerve cells. If we take a structure of neuron, we will have cell body. In the cell body, we will have some granules which are called as Nissl granules, which are mainly involved in the synthesis of proteins and neurotransmitters, which will be packed in the form of which is transported to the axon terminals, where they will be stored in the form of secretory vesicles. So Nissl granules, which are involved in the protein synthesis, which are present in the neuronal cell bodies, because they are involved in protein synthesis, they will contain more number of rough endoplasmic reticulum. And similarly, like there is one more example, acina cells of pancreas. You all know pancreas has endocrine pancreas as well as an exocrine pancreas. The exocrine pancreas has acina cells, which is mainly involved in the pancreatic juice secretion, which has many enzymes which are involved in the digestion of proteins. So they have examples, the enzymes like trypsinogen, chymotrypsinogen, all these are also proteins. So acina cells of the pancreas are also contain more number of rough endoplasmic reticulum. So these are the examples for rough endoplasmic reticulum. And smooth endoplasmic reticulum, it is present in cells which are involved in the steroid or lipid synthesis. So Leydig cells, these cells are present in the testis. So testis, in the males, they will have two different types of cells, Leydig cells and Sertoli cells. So this Leydig cells are involved in the production of testosterone, which is actually a sex hormone, which is a steroidal hormone. So which is a hormone which is mainly derived from cholesterol. So Leydig cells, they have more number of smooth endoplasmic reticulum. And cells of adrenal cortex, we have the adrenal gland, which is divided into adrenal cortex and medulla. This adrenal cortex has, which will secrete more sex hormones, and again because of that function, adrenal cortex will have more numerous smooth endoplasmic reticulum. So rough endoplasmic reticulum, it is not only involved in synthesis of proteins, it is also involved in folding of proteins, and this is done by special type of proteins which are named it as chaperones or heat shock proteins. What is meant by chaperones or heat shock proteins? These are the proteins which are involved in the folding of proteins. So these are called chaperones or heat shock proteins. So the next cell organelle is Golgi apparatus. What is Golgi apparatus? It is collection of membranous vesicles, sacs or tubules. So what is the function of Golgi apparatus? Golgi apparatus is present very near to the endoplasmic reticulum. So once the proteins are synthesized in the endoplasmic reticulum, rough endoplasmic reticulum, they will be packed by the Golgi apparatus into the secretory vesicles. So any cell which is involved in the secretion, they will have more number of Golgi apparatus, like goblet cells in the intestine, bronchial epithelium and plasma cells. So the sorting and processing of proteins is done by Golgi apparatus. We can remember it as the packaging of proteins is done by Golgi apparatus. And glycosylation of proteins and lipids. We saw that there are different types of proteins, there are different types of lipids even in the cell membrane. We said when the carbohydrates are attached to the proteins, they are glycoproteins. When the carbohydrates are attached with the lipids, they are named it as glycolipids. So glycosylation of proteins and lipids is done or is also a function of Golgi apparatus. And formation of proteoglycans is also a function of Golgi apparatus. One more function is there which is very important. Lysosomal enzymes are synthesized in the Golgi apparatus. All these are the functions of Golgi apparatus. If you see the structure of Golgi apparatus, so as I told you, Golgi apparatus is present very near to the endoplasmic reticulum. So the endoplasmic reticulum synthesizes proteins, and these proteins are packed by the Golgi apparatus, and they will be present within the secretory vesicles, right? So here we can see clearly. So at the end which is facing the endoplasmic reticulum, that is called cis end, which is the receiving end, which receives the proteins which are synthesized from the ribosomes, and this end, this is called as trans end, which is the shipping side. So this end is the cis end, which is the receiving end, and this end is the trans end, which is the shipping end. So Golgi apparatus is also a polarized structure. So the next cell organelle is lysosome, which is also called as suicidal bags or residual bodies, which is considered as the recycle bin of the cell. So these are round membrane bound organelles. Lysosomes are the round membrane bound organelles. Lysosomes mainly contain the hydrolytic enzymes. They contain about 40 hydrolytic enzymes. So any cell which is involved in phagocytic activity will contain more number of lysosomes. For example, neutrophils and macrophages, which is mainly involved in the phagocytosis, right? Which requires more number of lysosomes. So here we can see when the bacteria approaches the neutrophil or macrophage, these neutrophils will extend a pseudopodias and they will be engulfed. This bacteria will be engulfed, and then these engulfed bacteria will be lysed by the lysosomes, and then the materials, the digested materials will be expelled out from the cell by the process of exocytosis. So this is how the phagocytosis will occur. So what is phagocytosis? Phagocytosis is engulfment of foreign bodies. That is the process which is called as phagocytosis. So the cell which is involved in the phagocytic activity is mainly neutrophils and macrophages. So they will contain more number of lysosomes, which is mainly involved in the destruction of these foreign bodies. So what are the functions of lysosomes? It forms the intracellular digestive system, which will digest damaged cellular structures, which will digest the food particle which is ingested into the cell, and it will also lyse the bacteria. So these peroxisomes, their function is they have two important functions. One is detoxification of the harmful substances, and the second important function is they synthesize and they degrade the hydrogen peroxide, H2O2, which is the hydrogen peroxide, which is a reactive material which is degraded by the peroxisomes. These two are the very important functions of peroxisomes. So any cell which is involved in detoxification will have more number of peroxisomes. So one of the function of liver is detoxification. So liver cells, which are called as hepatocytes, they will have abundant number of peroxisomes. So centrosomes, which is the last cell organelle, it consists of two short cylindrical structures. Here we can see there are two short cylindrical structures which are called as centrioles, and this centrosome is responsible for the movement of chromosomes, which is located usually near to the nucleus. So that's all about the cell organelles. So mitochondria, endoplasmic reticulum, then we have Golgi apparatus, then lysosomes, peroxisomes, centrosomes. So all these are examples for all these are the cell organelles, and we know the functions of cell organelles. In addition to the cell organelle, cytoplasm also contains cytoplasmic inclusion bodies. Sometimes these inclusions are present in the cell and they may not be present in some cells. The examples for this cytoplasmic inclusion is glycogen, which is one of the stored nutrient, right? Whenever there is excess carbohydrates, they will be stored in the form of glycogen in the muscle as well as in the liver and adipose tissue. So this may be present. If we do some exercise or physical activity, the glycogen may be utilized. If there is starvation, this glycogen may be utilized and it may not be present in the cell. So it is one of the cytoplasmic inclusion. The pigment granules like melanin, it may be present or it may not be present in the cell. So these are called as cytoplasmic inclusion. These are also present in the cytoplasm. So next structure which is also present in the cytoplasm is cytoskeleton, which is also considered as bones of the cell. So what is cytoskeleton? A system of fibers that not only maintains the structure of cells, which also permit the cell so to change the shape and to move. So it helps to maintain the shape, and it also helps to move the cell from one place to the other place. So it may be within the cell, or the cytoskeleton provides the strength, support, locomotion and intracellular transport and the spatial organization of the cell organelles. All these are the functions of cytoskeleton. So we told it is a system of fibers. So cytoskeleton consists of microfilament, intermediate filament and microtubules. And here we have a picture to show. Here we have the microfilament. This is the intermediate filament, and here we have microtubule. So microfilaments are mainly made up of proteins which are called as actin and myosin. Microfilaments are mainly made up of actin molecules. The diameter of the this microfilament is 7, which is the smallest diameter fiber. And the intermediate filament fiber, the diameter is intermediate, which is 10 nanometer, and it is made up of different proteins. Microtubules, they are especially made up of tubulins, there are two different types of tubulins, alpha and beta tubulins. It is a cylindrical structure and it's a hollow, long tubular structure, and the length of the diameter of this microtubule is 25 nanometer, which is the largest diameter. So microtubule, it is a long, hollow tubular structures. It is made up of two globular protein subunits, which are alpha and beta tubulin, which gives structural strength to the cells. And microtubules are also involved in the locomotion of the cells. They are especially present in the structures like cilia and flagella, which helps to the motility of the cells. So in our body, in human body, we have cilia, which is especially present in the respiratory mucosa, which helps to propel the dust particles, and it prevents the entry of the dust particles into the respiratory tract. And flagella also has some of the flagella also composed of microtubules, which also helps in the motility of the flagella. Intermediate filaments. So these are the most abundant filaments which will connect the nuclear membrane with the cell membrane, and it forms the flexible scaffolding for the cell, which helps in resisting of the external pressure. So absence of these intermediate filaments can cause easy rupture of the cell. Any abnormality in these intermediate filaments can cause blistering of the skin. Next cytoskeleton is microfilaments, and these are long solid filamentous structures which is especially made up of actin and myosin, which are the contractile proteins. And the actin is the most abundant protein in the mammalian cell. These microfilaments, they are also present in the microvilli, which are the extension of microfilaments. So that's all about the cytoplasm. And the last structure in the cell is nucleus. So here there are some cells which don't have nucleus. The example is RBC. Matured RBC is the non-nucleated cell, whereas some cell will have multi-nucleus. So skeletal muscle is one of the example for multi-nucleated cell. And here we have a typical nucleus. So the nuclear membrane, and then we have nucleoplasm, and then we have the nucleoli. So this nuclear membrane is similar like cell membrane. It is also a lipid bilayer membrane. The very important difference between cell membrane and nuclear membrane is the nuclear membrane will have more number of pores. You can see here there are many pores in the nuclear membrane, and the outer nuclear membrane is again outer membrane, and it has inner membrane. And the nucleoplasm will have chromatin material. When the cell is not dividing, we will have thread-like materials in the nucleoplasm, and that is called as chromatin. So here you can see another picture to show the nuclear membrane. We have outer membrane and inner membrane. The outer nuclear membrane continues with the membrane of the rough endoplasmic reticulum, and the nuclear membrane have nuclear pores. And in between the outer and inner membrane, we have a perinuclear space. So here we can see the chromatin, which is thread-like materials when the cell is not dividing. So when the cell is not dividing, in the nucleoplasm we can see thread-like materials which are called as chromatin. But when the cell division occurs, we can see rod-shaped structures which are connected at the centromere in the center, which has X-like configuration, which are called as chromosomes. And in the nucleoplasm we will have many number of chromosomes, which is the one which actually contains many genes. So the main function of nucleus is, it is the one which is especially important for reproduction. So nucleoli is present in the nucleus, and it is the site of synthesis of ribosomes. So thank you.